The overall goal of this procedure is to visualize DNA replication on the single molecule level. Begin by incorporating halogenated nucleotide analogues into newly synthesized DNA in living cells. Spot the cells with labeled DNA onto a microscope slide. Then, lyse the cells and stretch out the DNA onto the microscope slide. Subsequent immunostaining of DNA fibers and analysis of the fluorescent microscopy images can illuminate the global replication fork dynamics to allow quantitative analysis of parameters that influence the overall replication program.
Over the past years, different versions of the DNA fiber fluorography techniques were developed to visualize the movement of individual replication fork within living cells. This in vivo experiment in DT40 cells that you are just about to witness can be accomplished within a day and requires only general laboratory equipment and a fluorescence microscope. Demonstrating the procedure will be Dr. Rebecca Schwab, a postdoc from my laboratory.
In order to label DT40 cells in vivo, start with an exponentially growing culture. Add IDU label to a final concentration of 25 micromolar and mix the cell suspension well. Incubate the cells for 20 minutes at 38 degrees Celsius and 5% carbon dioxide. Next, add the CLDU label to a final concentration of 250 micromolar. Mix and incubate the cell suspension. Now wash the DT40 cells with ice-cold PBS. Resuspend the cell pellets in cold PBS and keep the labelled cells on ice. Spot 2 microliters of the cell suspension onto one end of the glass slide. Air dry until the volume of the drop is greatly reduced but not completely dry. Now add 7 microliters of fiber lysis solution on top of the cell suspension and gently mix the solutions by stirring with a pipette tip. Incubate for 2 minutes. Next, tilt the slides to 15 degrees to allow the fibers to spread along the slide. Once the fiber solution has reached the bottom of the slide, place it horizontally to air dry. At this point, a thin, opaque line should be visible along the slide. Mark the beginning of the stretched fibers with a pencil. First immerse slides in 3:1 methanol to acetic acid for 10 minutes. Wash the slides in distilled water, then immerse in 2.5 molar hydrochloric acid for 80 minutes. Wash the slides 3 times in PBS for 5 minutes. Dab collect excess PBS onto a paper towel, then orient the slides horizontally. Now pipette 5% BSA in PBS on top of each slide and place a cover slip. Incubate for 20 minutes. Move the cover slip gently down the glass slide. Remove the excess BSA with a paper towel. Then pipette 50 microliters of the anti-BRDU primary antibody solution onto each slide. Cover again with a cover slip and incubate in a humidified chamber for 2 hours. After removing the cover slips, wash the slides 3 times in PBS for 5 minutes. Apply 50 microliters of the secondary antibody solution. Position cover slips and also protect the slides from light. Then incubate for 1 hour. Remove the cover slips and wash the slides 3 times in PBS for 5 minutes. Finally, add a drop of Vector Shield mounting medium onto each slide. Gently press on a cover slip and remove excess fluid around it with a paper towel. Seal the cover slips with transparent nail polish and air dry. Store the slides at -20 degrees Celsius. Place a drop of immersion oil onto a slide close to the pencil mark and start locating the fibers. Move away from the main bundle to find areas where fibers are clearly separated from each other. In a typical experiment, select pictures only using one color channel in order to avoid bias. Then, take approximately 10 pictures of each sample. Move along the slide to take the different pictures, as one area of a slide may not provide representative fibre lengths or replication structures. Import pictures into an image analysis program. Measure the lengths of the 100 fibre tracts and/or count 150 to 200 different replication structures. The double labelling fibre technique allows one to distinguish between different replication structures. Newly replicated DNA can be visualized as lines of antibody-labeled nucleotide analogues. Here, an ongoing elongating fork is represented as adjacent red and green signals. New initiation events can be divided into origins that have fired while the cells were incubated with the first label and origins that have fired during the incubation with the second label. The former consists of neighbouring green-red-green signals and the latter of a green line only. Termination events manifest as adjoining red-green-red signals. Interspersed origins consist of consecutive origins and termination signals. Depending on the experimental design, stalled or collapsed forks can either be defined as a red-only signal or a red line followed by a short green tract. In this experiment, the wild-type DT40 cells have an average fork speed of 0.4 microns per minute with 63% ongoing forks, 10% origins, 16% stalled forks, 8% terminations and 3% interspersed fibres.
After watching this video, you should have a good understanding of how to visualize and analyze replication fork dynamics in DT40 cells. This will provide you with an essential tool for investigating defects in DNA replication.
Take into consideration. This method allows for quantitative analysis of parameters that influence DNA synthesis during S phase at a single molecule level. A crucial step to achieve this is the incubation time of the cell suspension on the microscope slide as well as lysis time. The practical part of a DNA fiber experiment is typically accomplished within 1 day. However, subsequent picture acquisition and analysis is more time-consuming and requires practice. Take into consideration. The labeling protocol can be adapted to answer various scientific problems, using an agent that interferes with replication during the second pulse labeling monitors fork elongation/stalling in response to replicative stress. In this scenario, the first label does not need to be washed out as the second nucleotide is added in excess. Alternatively, drugs that impede replication can be used in combination or just after addition of the first label. This requires the first label and the drug to be removed before the second nucleotide analog is applied. This procedure allows one to determine the importance of various DNA repair factors on replicative fork stability/recovery under conditions of replicative stress, i.e. fork stalling and/or collapse. Thank you for watching. Subscribe if it is useful.